The third and final presentation in this series is not going to focus so much on emotion, but is going to focus a lot more on memory. And it's actually a phenomenon in your book on page 524. We're going to talk about a physiological phenomenon called long-term potentiation. There's a ton of details about this process, but I'm going to try to just make it pretty simple and basic to you to kind of give you an introductory understanding of what the basis is for long-term potentiation, and then we'll figure out the implications from that. The idea is when you're doing long-term potentiation, many times you work with rats, number one, and what you actually have to do is you actually have to get cells from the rat's brain. In particular, if you're talking about memory, what they do in a lot of these studies is they're able to actually get slices out from the hippocampus. So they get cells out of the hippocampus of the rat. They put them in a little petri dish here so they can do various things to these slices from the hippocampus. There are a lot of different structures in the hippocampus. We're not going to go into detail there. Instead, what I'm going to do is explain the simplest type of long-term potentiation that was discovered actually now almost 30, 40 years ago per se. What they basically do, so they basically take a synaptic area. So there's an area in the hippocampus where there's a presynaptic area, and then there's a postsynaptic area. So what they basically do, maybe in stage one of this, is they just take this presynaptic pathway, or this, synaptic pathway, has a presynaptic area, and then there's a synapse in there, and then the postsynaptic area. They take this synapse, and what they do in phase one is they start it off and they put a little bit of electricity in there. So the idea is you put just a tiny bit of electricity, you do a little electrical stimulation, simple stimulation, into the presynaptic area. And what you basically find is if you put a little electricity in presynaptically and then you measure the electricity going on postsynaptically, what you basically find is just a little energy postsynaptically. So it's basically put a little electricity in presynaptically, and then the output is a little electricity postsynaptically. So that's phase one. In phase two, into this hippocampal pathway from slices, what you do is you actually put something called tetanic stimulation in. Tetanic stimulation is basically high voltage electricity into the pathway. And so the idea is it's called tetanic stimulation. You put a whole lot of electricity into these slices presynaptically, and then of course if you measure postsynaptically, you see a very large electrical response, and if you remember back from our previous units, it's called a large EPSP, is what you see, a large excitatory postsynaptic potential. So it's a very simple idea. A little electricity in phase one, a small EPSP on the output of phase one. Phase two, you put a lot of electricity in, and then you measure the output postsynaptically, and you see a lot of electricity in postsynaptically. Phase three is the very interesting phase. What they've actually found is after you do this for phase two, if you go back in during phase three and just deliver a little electricity in, now if you remember in phase one, a little electricity led to a little electrical output, but if you do this phase two thing first, what seems to happen in phase three is you put a little bit of electricity in, just one simple stimulus, and you seem to get a huge EPSP on the output side. A huge EPSP on the output side. And so what researchers have named this is long-term potentiation. It seems that phase two potentiated, made it stronger, the particular pathway, and now just putting a simple electrical stimulus in is causing the same response. Now, the little electrical stimulus in phase three, that might happen 1 hour after the tetanic stimulation, but it can actually happen up to almost 100 hours later. So if you do phase two, the tetanic stimulation, and then measure the output, you see a lot. And then if you go in even up to 100 hours later, it seems, and just deliver this single stimulus into the same pathway, you get a huge EPSP. And so that's the phenomenon known as long-term potentiation. And the reason this has captivated a lot of people's interest is people think this might actually be the basis of memory. The other thing I should add, back to what we just talked about, it seems the neurotransmitter that is very involved in this LTP is the neurotransmitter glutamate, which actually makes some sense even with Alzheimer's disease. We know glutamate is involved with that, so glutamate seems to be the key neurotransmitter involved here. So the reason people are so excited by this is people think this could be what's going on when you memorize information cognitively into long-term memory. They think maybe this is what's going on when you're memorizing information. So let me give you a little example of this. Let's say you're studying for a Mary Baldwin test for next week, and let's say, just as a cognitive analogy, kind of a mind-body thing, let's say the cognitive analogy is you study a whole bunch. So if you take this idea of studying a whole bunch as almost tetanic stimulation, you study a whole bunch and you try to memorize as much as you can. Maybe that is the tetanic stimulation going on inside your brain. And then you get to the test, and what does the test have? Simple question. So that could be like the simple stimulus in phase 3. You get a simple question, and let's say you do well on the test. The simple question triggers all your memories. So you're able to recall all that information. Maybe that's like phase 3 where you get a simple electrical stimulus and you get the whole EBSP thing going on there. So cognitive psychologists really like this idea of how we memorize information, and this is actually an example of research in the field of cognitive neuroscience. Cognitive neuroscience. How information cognitively is mapping to brain neuroanatomy. We think that maybe when people memorize information at a cognitive level, maybe this LTP phenomenon is the thing going on in the brain. So all three presentations have this in common. Let's look at a little memory. Let's look at some emotional responses. Let's figure out what the conditioning is and the conditions to allow that. And then let's try to look at the neurobiological roots of this. Now, there are a lot of people with long-term potentiation. That's a physiological phenomenon. They're not sure if this really is what's going on when we memorize information or not. So there are a lot of critics out there, and that could very well be true per se. But the idea here, long-term potentiation, is the physiological phenomenon of delivering tetanic stimulation into the hippocampal pathway, which we know is involved in memory, and then at a time later, even up to 100 hours, you can go and just deliver a simple pulse of electricity there and get a huge EPSP. So that's called long-term potentiation, and people think this may be what's going on when you memorize information. That cognitive analogy that I drew between studying for a test and then getting a test question and recalling it. We have no direct proof of that analogy, but hopefully that analogy makes some sense to you as an idea, and this is in the field of cognitive neuroscience. So thank you very much, and that will conclude the third presentation.